Diabetes Connections is supported by Omnipod. For many of you, as you listen, this is going to sound like a weird question. Why do you use the insulin pump you've got on right now? There's a really good chance you went on that pump 15 or 20 years ago because it's what your doctor had back then, and you've never really thought about changing. That was us until Benny's pump company went out of business, and I was amazed at how much things had changed when we looked for a new one. I'm telling you, it is time to take advantage of what's new. If you need a sign to switch, this is it. Omnipod 5, the tubeless automated insulin delivery system, is here, and it's available through the pharmacy, which means you can try it today, even if your tubed pump is under warranty. Fill out the quick online form, see if you're eligible for a free trial at diabetes-connections.com. Click on the Omnipod logo for full safety risk information and free trial terms and conditions. Also visit omnipod.com/diabetesconnections.
This is Diabetes Connections with Stacey Sims.
This week, checking in with Sernova and their progress developing a cell pouch system as a functional cure for type 1 diabetes. Sernova's CEO, Cynthia Pussinen, joins me with an update on their latest research, and she answers your questions about everything from immunosuppressive drugs, how long people in these trials stay off insulin, and how to get into their trials. This podcast is not intended as medical advice. If you have those kinds of questions, please contact your healthcare provider. Welcome back, everybody. I am your host, Stacey Sims. You know we aim to educate and inspire about diabetes with a focus on people who use insulin. And Sernova is one of the companies hoping that their implantable stem cell system will be a functional cure for diabetes and help people stop taking insulin. We last talked to them in the summer of 2022 when they had announced that the first person in their trials with type 1 was completely off injected or infused insulin and had started making their own. I wanted to talk to them again and see how everything is going. The transplantation of islet cells is an idea that has been around for a very long time and has been used with varying amount of success by different companies. We've talked about this a lot. I'm not going to rehash it all here. I will link up lots of episodes we've done on the past if this is your very first time hearing about it. But the basic idea here is that even if you implant healthy cells that can start making insulin, the body will eventually attack those cells. So people who have gone through this procedure need to take immunosuppressive drugs. Companies like Sernova are trying to get around this by using something like a cell pouch that not only protects the cells, but as you'll hear, really creates kind of a small ecosystem that the body begins to recognize as really just part of itself. There are a few people who have gone through these trials for Sernova, and we will talk about that and their success, but they are off insulin, what they call insulin independence. And we will talk about exactly what that means and what the hope is here for a much larger population. My guest, Cynthia Pussinen, became CEO of Sernova last September. She had most recently been the CTO, the Chief Technical Officer for Spark Therapeutics, which is a gene therapy company. She also has family members with type 1 diabetes, and she talks about that. You'll hear our whole conversation right after this. It's here. The all-new Dexcom G7 continuous glucose monitoring system is now available. Powerful and simple, Dexcom G7 is proven to lower A1C and help you spend time in range, all without finger sticks, scanning, or calibrations. There are so many amazing new features to talk about here. I'm not sure where to begin. Dexcom G7 is 60% smaller than the previous generation and is the most accurate CGM system out there. The 30-minute warm-up period is fantastic. It's up to 2 times faster than any other CGM brand. And I love the all-in-one sensor and transmitter. To learn more, go to diabetes-connections.com and click on the Dexcom logo. Cynthia, welcome to Diabetes Connections. Thanks so much for spending some time with us today. I appreciate it.
Thank you so much, Stacey. I'm really looking forward to talking to you. It's wonderful to be together this morning and look forward to chatting about type 1 diabetes.
Yeah, and this is an exciting time. Last time I spoke with anybody from Sernova, it was 2022. It was about a year and a half ago.
Okay.
Before we jump in with announcements and advancements, would you mind just resetting? What's the goal here? What does Sernova hope to do?
Sernova hopes to change the world at a very fundamental basis. The way I look at who we are and what we do and our reason for being at Sernova is envisioning a future where chronic conditions are no longer insurmountable obstacles. And our flagship program, our first program in the clinic, is for the treatment and truly the functional cure of type 1 diabetes. And so we're absolutely thrilled with the honor that we have placed upon us to be able to work with patients around the world and to hopefully change their lives in transformational ways and the lives of their families too.
I love hearing that. It just kind of gives you chills. So in a practical sense, this is islet cell transplantation. So tell us what that means in practical terms, what Sernova is doing.
At Sernova, we're taking an approach that marries up therapeutic cells. And so in the first situation in the clinic, we are using donor islet cells. So these are ethically sourced cells from donors. As you know, there are a limited amount of donor pancreases in the world. And so we recognize that we needed another approach, another future approach to treating patients. And so I'll talk about in a couple of minutes our induced pluripotent stem cells that we are engineering. But today we are taking those donor islet cells, we are placing them into a delivery device that we call the cell pouch, and then we put it into the bodies of patients, and it goes off and does the work that the body previously was unable to do, i.e. to do those functions that the islet cells in the pancreas were supposed to do. But with type 1 diabetes patients, as you know, those islets are missing, they're dead, they're gone, and that's not a type of cell that regenerates. So our approach with the cells in the cell pouch is to function as that cure, that functional cure for type 1 diabetes patients.
You mentioned donor pancreas. When people talk about stem cells, would you mind just giving us more of an overview? What makes that different?
There are a couple of different approaches that you can take when you're using cells in treating patients. So our first approach to the clinic is using donor islet cells. And so these are sourced from donors who leave their pancreases to medical science. There are companies out there who are also using donor islet cells. There are companies out there who are taking an approach to trying to treat type 1 diabetes with embryonic stem cells. We do not use embryonic stem cells in either the clinic today or in our future plans. For us, this is really important. We wanted to ensure that we are taking an ethical approach to everything we're doing. And we recognize that the use of embryonic stem cells is a bit of a tough topic for many people. And many European countries, in fact, will not allow embryonic stem cells into the country. And where we will go with the US elections, who knows? But that might be another topic that becomes challenging for the US as well, right? And so today we're using the donor islet cells. The reason why we started out with donor islet cells is because it was an available option to us, and it was a way to define or to establish that proof of concept that our idea of cells coupled with the delivery device, the cell pouch, can work. And in fact, it works so beautifully, Stacey. We have 6 patients right now who are insulin independent. And I'm going to read you a quote in just a moment from our first patient in our clinical study, because it's absolutely astounding. And you know what this means to the lives of patients and to their family members. But in the future, what we'll use is induced pluripotent stem cells. And so it's an engineered source of cells. And it's really interesting. I don't know if you know how stem cells work, but they are just kind of like futuristic almost. And so you can take a blood sample and you can engineer it or build it into any one of the 22 cell types in the body. Wow. And so I can make with a blood sample skin cells, I can make liver cells, I can make anything that's in the body, thyroid cells. And in this case, we're making islets, right? So those islets that are in the pancreas, you can write a protocol. So you take that blood sample, you write a protocol that helps you to build those cells or engineer that blood sample into the cells that you want. And then it typically takes about 27 days to take that blood sample and then generate that cell that you want. In our case, those islet cells. Yeah. So we can do in 27 days what it takes the human body 9 months in utero to do, to build those cells. And it's just very cool. I'm a science lover. And so I look at this and I just think it's so much fun to be able to consider the art of what's possible and where could this go. And, you think futuristically and about generating organs from a single blood sample, right? So that's just really cool for me.
I'm going to let you read that quote from the patient. I just want to say, after you do, I want to come back and talk about the challenge here because it sounds amazing. Obviously, this is not easy, but I don't want to skip ahead. Tell me what the patients in the trials are saying.
Okay. This is a quote from our first patient in our clinical study. So she is now just about 4 years insulin independent. So she says, after completing the safety, tolerability, and efficacy study of Sernova's cell pouch for clinical islet transplantation, and as the first transplant candidate, I can easily state how absolutely wonderful life is to be free of always thinking of how to manage my diabetes. After having type 1 diabetes for 47 years, with approximately 21,500 injections of various cow, pig, synthetic insulins, 34,000 plus finger sticks, 1,500 urine tests, 15 years on the pump, carb counting, blood tests, low blood sugar reactions, and doctors, and more doctors' visits, I have now been free of the need of injectable insulin for 15 months. So this was back in 2021. She's now just under 4 years insulin independent. She finishes off to say that my only wish is that this could have been done sooner. Isn't that amazing?
It is amazing. And I have so many emotions listening to that. Let's revisit the emotions maybe a little bit later. But to clarify one thing, when you say insulin independent, so no basal, no bolus, no insulin.
No insulin.
I guess we should say no exogenous insulin. That's the right way to say it.
No exogenous insulin is probably the best way to put it.
Just the insulin in her own body.
Yes, exactly. So the cell pouch that we have that we implant into the patient's body, once we load that with the cells, the donor islet cells today, it essentially becomes a pseudo organ. What you've got in your body is essentially a pseudo pancreas doing the work that your pancreas should have been doing, and that the pancreas of those of us who don't have type 1 diabetes does do, which is amazing.
Okay, let's talk about some of the challenges here because it is amazing to think about how this works. But when you're talking about these donor cells and even these stem cells, right? They're not the person's own. They're not that woman in the study. They're not her own. So her body is trying to fight them off. Exactly. This is the big problem of rejection. Where do we stand with that? Right back to our conversation in just a moment, but Diabetes Connections is brought to you by Gvoke HypoPen. Gvoke HypoPen is a ready-to-use rescue pen for treating very low blood sugar in people with diabetes ages 2 and above. If you take insulin or sulfonylureas, you're at risk for your blood sugar going too low. Low blood sugar emergencies can happen unexpectedly, and they demand quick action. You need a safety net when it matters most. Be ready to treat very low blood sugar with Gvoke HypoPen. Find out more, go to diabetes-connections.com and click on the Gvoke Glucagon logo. Gvoke shouldn't be used if you have a tumor in the gland on top of your kidneys called pheochromocytoma, or if you have a tumor in your pancreas called insulinoma. Visit gvokeglucagon.com/risk for safety information. We right now in the clinic have to use immunosuppressive regimens. So in our first cohort of patients, which has 6 patients in it, 5 of those patients are insulin independent, and the 6th one came a bit later in the study. So he or she is not, we've not yet pulled the data on them. So they are being treated with immunosuppressive regimen. That immunosuppressive regimen included tacrolimus and MMF. That's a tough combination, isn't it? So tacrolimus is not easy for the body. When we went on to our second cohort of patients, which has 7 patients in it, 6 are in the study now. We have 1 other patient to recruit, and then that cohort will be done. One of those patients is insulin independent also. Haven't gotten the data on the other 5 yet. So what we did was to modify that immunosuppressive regimen. So we use a far lower dose of tacrolimus, and we're also using belatacept. And so we found that that is much better tolerated by the patients. And, immunosuppressive regimens are tough. I have another chronic condition where I take immunosuppressives, and it's hard. It's hard on the body. It's not something that you want to do. And as a patient, you just find that you're stuck doing that in some cases. But what we are doing, Stacey, at Sernova is we're also looking at how can we answer that question? How can we obviate the need for immunosuppressive regimens, because we don't like them. Patients don't like them. Doctors don't like them. So we have this technology called conformal coating. And what conformal coating does is it essentially acts like a shrink wrap around the cells that we put into the body, so that when your body sees those cells, it doesn't think invader, and then launch that immune attack on the cells.
So we are really hopeful that the development continues in a positive manner on this work that we're doing, because you think about what that could also translate to beyond type 1 diabetes, in fact. But that could be used for multiple other conditions. And so the impact there could be rather significant. But for type 1 diabetics specifically, we can cloak those iPSCs that we're engineering now. We could cloak those donor islet cells that we're using today, and then you wouldn't need the immunosuppressive regimen. So it is our goal to provide a solution that does not include an immunosuppressive regimen moving forward.
Where do you stand with the coding? Is that something that is in people yet? Where are we in the trials?
It's not in people yet. So we do have it in development. So we are working with the University of Miami. We have a partnership with them on this. And so the conformal coating is essentially a biocompatible cross-linked polymer. And that's the stuff that forms a shrink wrap coating around those therapeutic cells that then get put into the cell pouch chambers. And so the coating, importantly, provides for that normal insulin and glucose kinetics. It provides for or enables that transfer of oxygen and nutrients as well. So your islets do survive quite nicely. So we're at the point right now where we are scaling up production of that conformal coating. So up until this point, we've been doing it by hand. And so you can imagine like trying to put this coating on by hand around the cells. It's just labor intensive. So what we're preparing and what we're almost complete with is a machine, essentially, that does it in an automated fashion. So that system design is complete. The piece of equipment itself is almost complete as well, and that'll include the entire coating process, and it'll be done in a GMP compliant manner, which is a huge plus. So stepping back for a moment and completing the answer or the response to the question that you asked, where are we with conformal coating? So we've done a number of preclinical studies, and what we're able to show is that those conformally coated islets in the implanted cell pouch achieves that normal blood glucose control in type 1 diabetes. So this has been done in animal models. We're going to continue to do it in a few other animal models because, as you know, when you go to the FDA to get an IND for use in patients, you've got to show or demonstrate that you've done a number of animal models, animal studies. And so that's what we're continuing to do right now, is to just finish up those animal studies that we need to do. My hope is that when we go into the phase 1-2 clinic with our iPSC cells, that we can start it out immediately with the conformal coating technology. If we don't, what we'll do is probably 2 or 3 cohorts of the study or 2 or 3 portions of the study like we're doing now. Start out with just iPSCs, and then in the next cohort, do iPSCs plus that conformal coating technology.
I have some follow-up questions on that, but I want to go back to something you said that just really, I had this strange visual when you said we've been coating this by hand. I imagine like a guy looking in a microscope. I mean, this stuff is tiny, right? I mean, how are you coating it by hand? It's not like you're painting a fence.
It's so labor intense. It really is. And so essentially, you've got these groups of cells. And so just for range of understanding, the amount of islets that we're putting into the cell pouch and that amount of islets that you have in your body is only about 2 tablespoons worth. It's not like a liter of cells. So it's a really small amount. But with that conformal coating, you've got to make sure that you're coating all of the cells equally and that you're offering that protection to each one of the islets. So it does take time. So for all of the animal studies we're doing, which have been a fairly significant number of it, we're spending a lot of time coating those cells. So it's, looking through the microscope and making sure that we're putting or applying that coating uniformly to each one of the cells. So once we're able to get that GMP machine in place, it's going to save so much time and will just be done in such a more efficient fashion. But you're right, it's essentially looking through the microscope and applying that coating.
I got some listener questions I wanted to ask, and one of them is from Maddie, and she wanted to know if you'd thought about where in the body the pouch is most efficient. Like where does it go, and is there a place that works better than any other?
Yep, there absolutely is. And so we've done a lot of work on this with our current clinical principal investigator. So his name is Dr. Peter Witkowski from the University of Chicago, and he's a world-known expert in the field of transplantation. So we're working with a really good medical institution and principal investigator. And he's helped us learn quite a bit about the placement of the cell pouch as well as the dose and then the cell density of those cells. So the cell pouches are contained or placed within the abdomen area, and they are located in the subfascia, so kind of deep within the belly region, around the level of muscle. As a patient, you do not feel it in there at all. So the cell pouch is about a credit card sized shaped device. It's only a few millimeters thick. It's made of a polypropylene material. So this is a material that has been in patients, millions of patients around the world, in other medical devices. So it's borne out as safe. There's no issues with it being in the body. In fact, we have a patient who was on our very first clinical study, which was done way back when with the University of Alberta. And so this patient has had the cell pouch in their body for, I think, 14 years now.
Wow.
And it's safe. There's no fibrosing associated with that cell pouch. The patients say that when you've got it in your body, you don't feel anything moving around. You don't even know it's there. And so patients we have on the clinical program now do not want those cell pouches taken out because they're working so well. They want to keep the cell pouches with the cells in because they're essentially leading a more normal life, as it were, today.
Wow, that's really interesting. This might be a dumb question. I feel like I should know the answer to this, but maybe someone's listening for the first time. When the pouch is implanted, and maybe this is a theoretical question as well, you were talking about the longevity. Does it ever come in and out?
Unless there's a need for it to come out, no. So we envision that pouch staying in, and the cells should not ever need to be replaced. Because you think about the body of a normal non-type 1 diabetes individual, by the time you're 11 or 12, or maybe a little bit later, around the time you hit puberty, you have all of those islet cells that you are ever going to have. And so what we see happen with type 1 diabetes patients is that for some reason, those islet cells die off, whether it's an autoimmune reaction, whether it's some sort of hereditary reason, whether it's a response to a virus. We don't really know what the cause or what causes type 1 diabetes, but for whatever reason, the islet cells end up dying off in type 1 diabetes patients. And so with the cell pouch and the therapeutic cells that we put in, we don't feel that you will ever need to have a new cell pouch put in with the new cells. But if you ever did need to, we can do that. And it's as easy as a 15-minute procedure and the placement of a new cell pouch in the body. So our thinking is, and what the clinical experience has borne out thus far, and we have up to 4 years data in patients, we haven't needed to replace any cell pouches. We haven't needed to replace any cells. They are continuing to do the work that they did on day one. Which is really good news.
Yeah.
However, one thing that is important, and I'm going to tell you a story about one of our patients in our phase 1/2 study in the second cohort of patients. So as you know, we use the donor islets for this first study that we're in right now. And once we harvest those cells from the donors, we either use between 1, 2, or 3 donor pancreases in getting the right quality of cells to be transplanted into our patients in the cell pouch. And it goes through a GMP like cleaning and preparation process, so to speak, before we put it into the body. And once we put it into the body of the patient, we also send those cells off for different studies to make sure that there's nothing wrong with the cells. But we have to place them into the body immediately because they're living cells. And if we waited, then they would die. So with one of the patients, it was actually our first patient in the second cohort of the study, we found out that the donor pancreas had a candida infection. Oh. So candida is a serious fungal infection. And so we immediately took action. We explanted the 2 cell pouches that had affected cells in it. And it was just absolutely tremendous to see that there is no evidence of a candida infection in the body of our patient once those cell pouches were explanted. And so what that tells us is that the cell pouch is keeping very tightly whatever we put into it. And so you think about the importance of this. So some of the pharmaceutical companies out there today are taking an approach where they essentially dump some cells into the portal vein. Imagine what would have happened if those candida infected cells had been put into the portal vein. At that point, to get those cells out, you're talking about a liver transplant. And that's just a disaster scenario for any patients with chronic conditions, because we are not going to be at the top of the transplant list. So in my opinion, that's a highly flawed approach to treating patients, to just dump cells into the portal vein alone, because we just don't know what could happen with those cells. We don't know where they could go. Essentially, the FDA, I think, really wants to see an approach like we are taking at Sernova, where you have a delivery vehicle, the cell pouch in our situation, and that can be implanted into the patient. It can house the cells, and then if need be, it can be taken back out, and showing the proof that nothing has gotten into the body that shouldn't have gotten into the body. So that unfortunate situation with our first patient in the second cohort was not intended at all. We didn't plan for it, but we are so happy that we were able to see positive results from it, because another thing, that patient is insulin independent.
Well, I mean, that's wonderful. And it's great on your side, but it begs the question, how does something like that happen? I mean, should we be worried about that? I guess I can call it a supply, being contaminated? That's scary to hear.
Yeah, it's a little bit unnerving, isn't it? And so that's why we don't feel like using donor islet cells is a commercially viable solution. Because one, you've got the quality situation. And, when we did all of the right things in the testing of the donor islet cells. It's just the data didn't come back until a few days after we had transplanted. Number two, with the donor islet cells, we know that approach will not be sufficient to treat the number of patients that we want to treat. This was only intended at Sernova to be a proof of concept, to show that the cell pouch plus cells can provide that functional cure. What we are using as our next approach that will be the commercial or off-the-shelf approach is these engineered iPSCs. So with that, those cells are made in the lab. They are all being put through the same battery of release criteria before they are ready for use. They will be stored in vials in freezers around the world. They can be shipped wherever we need them. And they are all the same exact quality. They pass the same analytical tests to get placed into the vial and used in patients. And that's an endless supply of product. So when we think about, will there be enough product for patients? Because we all know what happens now. You go to the pharmacy and try and get insulin, and you're told oftentimes we don't have insulin, which is terrifying as a patient to not have that therapy that you need to stay alive. With the iPSCs that we're engineering in partnership with our strategic collaborator in Germany, it's a company called Evotech. They're designing and then producing those islet cells. That is the commercial, off-the-shelf, no limits to how many patients we can treat approach. So then we don't have to worry about the quality, because the quality is built in to those cells. You don't have to worry about the amount or number of vials available to patients, because it's endless. We start getting low, we produce more in the GMP manufacturing facility.
I appreciate the point. I'm going to push back a little bit. I mean, insulin is made in gigantic vats. There's no shortage. There may be a couple hiccups here and there, but I just want people, as you listen, I've heard here and there, maybe a supply chain issue. Somebody says we have to order it. I have not heard of any kind of insulin problem in the United States. So I know what your point was there, and I appreciate it, but to be clear.
And I guess I just say that from personal experience. When one of my nieces tried to get insulin and the supply wasn't there, she had to wait 4 weeks, unfortunately. So maybe it was just a glitchy thing where we live, and that for a patient is really scary. And thankfully, it doesn't seem like it's a widespread problem from what you're stating, Stacey. But I think the approach that Sernova is also taking is one that we don't ever want for patients to not have the therapy that they need.
Sure.
Because it is so functional. It is so foundational to being able to live a healthy and normal life. So we hope to provide that to as many patients who want it.
Got it. I know we're getting a little long here. A couple quick more questions for you. Another listener, Nick, had asked, and this was really interesting. Did the recent Lantidra decision impact regulatory environment in a positive or negative way for Sernova's product. And just real quick, I know you know Cynthia. Just real quick, Lantidra was approved by the FDA. It is the first cell therapy to be approved by the FDA for people with type 1, and it helps restore islet cells, is basically right what it does.
Yes. Yep. So I think that any progress that we make on the cell or gene therapy side of the life sciences business that provides additional information and learning to both the industry and to the FDA is a win, right? And so we have these first generation products, so to speak, for either cell or gene therapy. And then as an industry, we learn and we evolve and we iterate on anything that we see that comes out. And so that's exactly what we're doing at Sernova. We are evolving and we're learning more and more as a life sciences industry. And then in conjunction, the regulators around the world are also learning. And so I think what we're going to see is the bar will get consistently raised more and more as both the cell therapeutics space in life sciences learns more, as well as the FDA and other regulatory bodies around the world. Once they learn more, they're going to consistently say, okay, we need to see this, we need to see that, you've got to prove this piece of data to us, and that benefits patients. That is all exactly the things that we should be doing as an industry and as the FDA is consistently raising the bar on what it looks like to be safe and efficacious from the patient point of view. So, I think Lantidra was the first product out, first cell therapy product or solution for type 1 diabetes. And we're happy that it's out. We're happy that we're learning from it. And we're happy that the industry as a whole is continuing to iterate and evolve in a positive direction.
And then Claudia wanted to know, are you looking for clinical trial participants? And this is a question I get all of the time. How can people get into these trials?
Yes. And so I would say contact the University of Chicago because they are the ones who we're doing their clinical study through right now. When we start our next phase 1-2 study, what we will do is make certain to put that on our website and to push it out. And maybe we'll actually get back in touch with you, Stacey, to say, hey, we're opening up clinical sites, because my goal is to open a few sites in the US, one in Europe and one in Canada, so that we have more sites open and can treat more patients. And just one other comment, I showed that quote that I read to you a little bit earlier to my nieces who have type 1 diabetes, and their reaction was their eyes got big and they looked at me and they said, "Are you serious? This is for real?" And I said, "Yeah, it's for real." And one of them said, "Can I get on this study? Can you put me on this study, please?" So, patients hear that. And so back to what you were saying earlier, the emotions, this is real life. And this is so positively impactful. This is why I joined Sernova. So I hadn't heard of Sernova before I interviewed with the company 6 months ago. My role is new with Sernova. I've been here for just over 3 months as the CEO. And I've been in the life sciences industry for over 30 years now. And I should have known who Sernova is because of the tremendous data that we have. And when I started doing my due diligence on the role and started seeing the data, I couldn't believe it at first, that there are patients who are insulin independent with the treatment. And then the more and more I delved into it and started to see that the iPSC cells are on the horizon, that is the commercial go-to product of endless supply of cells for islet transplantation, I had to be part of it. We're here on this earth to make a difference in the lives of others, in my opinion. And I am so lucky to be able to work in an industry where we can change the lives of people in positive ways, in fundamental ways, and extend their lives and improve the quality of life. So thank you so much for talking.
Thank you. Oh,
I really so appreciate you, Stacey.
Well, thank you so much for coming on and sharing so much information. I would love for you to circle back when there's more information or ways to get folks into these trials. Let's keep the conversation going.
Yes.
Awesome.
I'll absolutely commit to it, and it's a date.
You're listening to Diabetes Connections with Stacey Sims.
Lots more information about Sernova and our previous episodes. You can just go over to diabetes-connections.com and learn more. You can also find those links in the show notes wherever you are listening, but I think the most comprehensive stuff is over on the website. Podcast apps can sometimes be a little bit hard to find, but you know the drill if you've been listening for any length of time. I know the progress on this stuff seems very, very slow, It's funny, we've been in this community for 17 years. My son was diagnosed 17 years ago last December, but I've actually been around the diabetes community for much longer. I've told this story before, but before Benny was diagnosed, I was the MC of the local JDRF golf tournament here in Charlotte, and I met a lot of the families. And I'll never forget that one of them, and I guess this was 2001, 2002, they had this team name or team logo, and it said Got Islets, and it was in the Got Milk logo. And that's when I first started hearing about islet cell transplantation. And it will be many, many years before this is something that many, many people can get. But I do know a few people who were in those early trials, and their lives are changed. These were people who had such severe hypoglycemia that they could no longer drive. They could no longer leave the house. They really couldn't even be alone. And islet cell transplantation has really changed things for them. The people I know personally are all taking insulin. This isn't what Sernova is doing, but it made diabetes livable for them in a way that would not have been possible. And when we think about it that way and you kind of break it up into, I don't know, maybe decades, I know it's frustrating that it's not coming faster, but I remain optimistic that we will look back and see this time as the beginning of something really, really exciting and different and changing the way diabetes is treated. I do hope this year to talk to some of the other companies involved in this technology. And of course, we'll keep you posted on Sernova's progress. I hope the new year is off to a good start for you. If you haven't already, please check out our new podcast, Diabetes Connections Type 2. It may not be for you, but I'm sure you know somebody with type 2 diabetes. So please tell them to check it out. It's the same concept that I've done over here, a news-focused show with actionable and interesting information where you get to ask questions of these companies and these CEOs. I'm really excited to be doing it for the type 2 community, and the response has just been so cool. It's been a lot of fun so far. We just started it in November. So please spread the word, Diabetes Connections Type 2, wherever you find your podcasts. All right, thank you as always to my editor, John Buchanan from Audio Editing Solutions. Thank you so much for listening. I'm Stacey Sims. I'll see you back here soon. Until then, be kind to yourself.
Diabetes Connections is a production of Stacey Sims Media. All rights reserved, all wrongs avenged.
As you're managing life with diabetes, Edgepark is here to support you on your health journey. Discover convenience and peace of mind with Edgepark, your trusted supplier in diabetes management supplies. Edgepark has a wide variety of pump and CGM solutions from top brands to fit your unique needs. Plus, Edgepark works with most insurance companies and handles the paperwork to navigate insurance complexities for you and make the ordering process as seamless as possible. You work hard to manage your diabetes. Let Edgepark take care of the rest. Visit diabetes-connections.com and click on the Edgepark logo to learn more.